Hey fellow lab rats, this is Rebecca from the Lab Rat YouTube channel. In this video I'm going to be demoing for you a gram stain procedure. Alrighty, let's get started. So today I'm going to be demoing how to perform the gram staining procedure. So gram staining involves a 4-step staining procedure, and it is the first test that is used in the microbiology laboratory to help identify unknown bacteria. So basically it helps to differentiate two different types of bacteria, gram-positive bacteria and gram-negative bacteria. Check out micro lectures on this channel, but in summation, so gram-positive bacteria have a thick wall of peptidoglycan outside of one plasma membrane, and this thick wall of peptidoglycan serves as a protection mechanism for that type of bacteria. Now, gram-negative bacteria have just two plasma membranes and a thin peptidoglycan wall. So the basis of this gram stain is whether the stain crystal violet is retained within the bacteria. So a thick peptidoglycan wall will retain the stain and cause the bacteria to appear purple. So this is the gram-positive bacteria. So gram-positive bacteria will appear purple in a gram stain. And a thin peptidoglycan wall will not retain that crystal violet stain and cause the bacteria to appear pink. So this is a gram-negative bacteria. So gram-negative bacteria will be pink, gram-positive will be purple. So I have a couple of bacteria here, and then I actually have my stains, and I'm going to go over these stains with you here before we get into the staining process. So this first one is the first stain that we use. It's a crystal violet stain, and this is a stain that gets into both gram-positive and gram-negative bacteria. Then we have grams iodine. So this is added to the sample, and it goes into the bacteria and forms crystals with that crystal violet stain. Okay, then we have grams decolorizer. This is usually a type of alcohol. So when added to the sample, it's only able to remove the crystal violet dye that is in the gram-negative bacteria because of that thin peptidoglycan wall. So this is how we get rid of the crystal violet from gram-negative bacteria. So after this decolorizer step, gram-positive will still be purple because that thick peptidoglycan wall retains the stain, but gram-negative bacteria will be colorless after this decolorizer is added to it. So after this step, so this is the third step, the fourth staining step is adding safranin to it. And safranin is a counter stain, and it stains anything that is colorless as a pink color. So that's how gram-positive bacteria are purple and gram-negative bacteria are pink color. Okay, so what we need to do here, again, I have two species of bacteria here for you today. I'm going to do a gram-negative and a gram-positive example. So this first one is Staph aureus. You can see it here. So this should be a gram-positive bacteria, and most specifically, it's a gram-positive cocci in clusters. So cocci are circular rather than rods, which are like rod-shaped. So this should be a gram-positive result. And then I have E. coli here, and this should give us a gram-negative result. Okay, now I also have two sterile slides here, and I'm going to take a wax pencil to label the slide. So I'm going to put negative, oh goodness, it's not very good, negative here, and positive here. And then what I'm going to do is I'm going to draw a circle about 1 centimeter in diameter in the middle of these slides, and that's where I'm going to put my bacteria. And that wax pencil is not doing anything for me today. It's giving me troubles. All right, so this is what they look like, all right? So what we need to do next is we need to take a sterile inoculating loop. So I have these sterile plastic loops. You can use a metal loop as long as you flame it. And I am going to drop a tiny drop of sterile water to the inoculating loop here. Okay, so I had a bit of an oopsie and I spilled water all over this, but basically what I want to do is drop a little bit of sterile water on here, and I'm going to put that in the center of that circle. All right, there we go. Goodness sake. Okay. Okay. And then I'm going to throw this away and use a different sterile inoculating loop for the next step. Okay, so I have a little bit of sterile water in between this circle here on both of the negative and the positive slide. So then I'm going to take another inoculating loop. And we know that the E. coli is a gram-negative, so I'm going to put that in. I'm going to transfer the E. coli to the gram-negative. So I'm going to take, again, another sterile inoculating loop, and I am just going to lightly touch one colony here. Lightly touch. You don't want to transfer a ton of bacteria onto the slide. Just tiny, tiny, tiny bit, right? And I'm going to rub that in that sterile water there. Okay? And then this gets discarded in biohazard. And then what I'm going to do is get another inoculating loop. Okay. Again, we want to have this sterile, okay, because we've already touched it to this bacteria. We've already touched one to this bacteria, so we don't want to transfer this bacteria onto this plate and on this slide. So this is going to be another sterile loop for our Staph aureus. And again, just barely, barely, barely touch one colony. And I'm going to put it in that area there. And then again, this is going to be discarded in a biohazard container. All right, so we need to let the sterile water evaporate from these slides and let it dry completely. I usually set like a timer for about 10 minutes or so. And then after it is dried completely, we heat fix the bacteria. And so this allows for the stain to work a little bit better on it. So that's the next step. After, I'm gonna let these sit and dry a little bit, and then I'm gonna put it on the heat plate to heat fix it. And once that is complete, I will go ahead and restart the video to show you actually the staining process. Okay, so I've let those gram stain slides evaporate the liquid and then put them on a heat block. Actually for quite a while, I kind of stepped away and kind of forgot about them, but they are here nevertheless. So what I'm gonna do is transfer these over to a designated gram staining sink. Let's see if I can make that there. That works. Okay, great. Okay, so I have these completely dried, and you can see there's like dried material in between the contents of that circle. So the first step that I'm going to do is take out my crystal violet, and I am going to drop it on the stain or on the circle there and let it sit for 1 minute. Okay, so after that 1 minute mark, I will return. All right, so my 1 minute timer went off, and the next step I'm gonna do is take some water and I'm gonna rinse that crystal violet off of there. That step. So at this point, both gram-negative and gram-positive bacteria will be purple. All right. Then the next step is I'm going to put some grams iodine on it. All right. And I'm going to set my timer for 1 minute. And when that timer goes off, I'll come back to you. Okay, that 1 minute timer has gone off. So I'm going to dump this iodine off of these slides, and then I'm going to rinse them with water again. Okay, what's the next step here? Do you remember? It's decolorizer. All right, so this is what's going to remove the crystal violet stain from the thin walls of the peptidoglycan layer on gram-negative bacteria. So what I like to do with these is I just set them at like a 45-degree angle, and I'm just going to cover them or rinse them with this decolorizer until the crystal violet comes off of it. You'll see what I'm saying. Okay, you see how there's just crystal violet coming off of the slide? All right. That should do me. Maybe there we go. Okay. Now you don't want to over decolorize. So a lot of times laboratories will have a specific time that you want to do it. So I'm rinsing them with water again, just FYI. All right. I believe most labs, at least the ones that I've been at, have a 10-second step for decolorizer, but just follow whatever your lab says. Okay, all right, so I've rinsed off the decolorizer, and then the very last step is using the counterstain, which is safranin. And the safranin, I'm gonna put on, okay, and then set for a 1-minute timer. So the safranin, again, is a counterstain, and that is what's going to stain the gram-negative bacteria with that pink color stain. Okay, after this timer goes off, I will come back to you. Okay, you hear my timer. Let's turn it off. And the last step here is rinsing off that safranin with this water. Oh goodness, I'm losing water. There we go. A nice rinse. Okay. So now what we're going to do is we're going to let these dry, and then once they're dry, then completely dry of course, we're going to look at them under the microscope. So I'm going to let these dry, and I'll see you at the microscope here momentarily. Okay, so this is my microscope view for the slide that we did on the Staphylococcus aureus species, and you can see very classically here, it's a very classic staph. Gram-positive cocci in clusters. There's some tetrads there as well, but you can see very clearly a gram-positive, and our stain looks great. Okay, so here's a good example of E. coli under the microscope with a gram stain. This is actually not the stain that actually the slide that I stained in the demo, but this one you can see a little bit easier. These are gram-negative rods, which is E. coli, or can be E. coli, but this one specifically is E. coli. It's actually in a blood culture example. But you can see the gram-negative rods, they are rod in shape, and then they also are pink, which means that they're gram-negative. Alrighty, hopefully this video helped you. If it did, go ahead and give it a like, and make sure to subscribe to my channel for more educational laboratory content. Until next time.